In this session, we will use bioinformatics applications to simulate gel electrophoresis analysis of restriction fragment length polymorphisms. The prep work that happened prior to this session are that we downloaded a reference DNA sequence for a gene. We then created a variant of that DNA sequence that either creates or destroys a restriction enzyme target site. In this session, we use a bioinformatics application by DNA Star to perform the simulated RFLP. So let's open GeneQuest and then open the reference sequence file for the CFTR gene. So we find the file, select it, and open it. And it displays the sequence and a variety of analyses. So we're going to go to the enzymes and scroll down to an enzyme that we focused on in an earlier exercise, XmnI. We drag that into the display pane. And we also need to change this to linearizing the sequence. Once we've done that, we can then simulate the results of a gel electrophoresis analysis. We can see how many restriction fragments there would be generated and their respective links. And we can also simulate what these would look like in a gel analysis. So let's drag that over so that we can have it available for comparison with the earlier part. We can see here the information that's displayed about the fragment links. So now that we've done that, let's open the second file in which we've introduced a variant that destroys the XmnI site. So that file is named as created in an earlier session. And we open that file, and again we repeat the process of adding the XmnI enzyme to the display pane by dragging it. And again we linearize the sequence. And then we can display the restriction fragment sizes, and we can see that there's one fewer restriction products, and then we can also display the simulated gel analysis. So let's try to tidy things up a little bit by putting our summaries of the restriction fragment products together, minimizing the other panes, and then displaying our two gel simulations side by side. Doing this, we can start to see where the banding patterns have changed as a result of losing the XmnI site. And in this slide, I make it a little bit easier to see that in the native DNA sequence, we have two fragments, one that's 513 base pairs and the other that's 201 base pairs. Those combine after losing the XmnI site into a fragment that is visible in the variant sequence, and that fragment size is 714 base pairs. And then when you compare the banding patterns, you can see the difference in results. This shows how this can be a very nice diagnostic test. So in this session, we've simulated a gel electrophoresis